Hi, I'm Annie Chai, and I'm from Biodiversity Research Center of Academia Sinica in Taiwan. Today, I'm going to talk about a fungi called Pyricularia oryzae, and especially from the perspective of evolutionary analysis. So the title is called Genomic and Evolutionary Analysis of Rice Pathogen Pyricularia oryzae strains isolated from rice fields. I have to start it with a plant disease called rice blast. It is a plant disease caused by a fungal pathogen called Pyricularia oryzae, which is the main role today. And this disease is threatening the world food security, resulting in up to over 30% of the food loss now around the world even nowadays. The relationship between the pathogen Pyricularia oryzae and one of its main attacking disease is rice, and the relationship is actually the pathogen-host interaction, and such interaction is the main driver of the adaptive evolution in the pathogen, and how and the relationship is based on a specific proteins called effectors. They are the main proteins that pathogen uses to attack the host, while in defense, the host develops the resistance. And such interactions is also called co-evolution. And such co-evolution between the pathogens and the hosts is serving as the main driver of genomic diversity within pathogens. So, to approach the aim of the Pyricularia oryzae evolution, we have to obtain the genomes more as possible, and based on the next and the third sequence technologies advancement, it makes it available to have the whole genome assemblies. And why we have to obtain more genome assemblies, that is because for Pyricularia oryzae, it has only one reference genome for now, called 1715, and it was firstly released in 2005 by shotgun sequencing. So in this study, to gain more insights on the genome composition of the pathogen fungi, we sequenced the two strains by PacBio sequencing, attempting to get completed genomes for approaching to get more insights on this in this study. And with a collection of the other 18 strains isolated in this study from rice fields across the time and space around the world, we expect that to gain more information on genomic aspects within the pathogen. So here, from the left figure is the main workflow that we conducted for this research. And from data preparation to the aims to for this project. In the first, we did de novo genome assembly instead of the reference guide genome assembly. It is because we want to prevent any bias of the reference. So we did the de novo genome assembly, and we tried the best to do the de novo genome assembly because it was not a easy process actually. And after that, we could obtain the genome annotation and then get the ortholog inference. And based on the ortholog inference, we can conduct the evolutionary analysis and try to find the effector gene gains and loss. So there are three main aims for this project. The first one is potential novel reference genomes. The second one is pangenome analysis, and the third one is about effector genes turnover. And the third one will not be talked about a lot in this poster at this time. But that is the the last point that we want to approach in the end. So here in the map, all of the strands in the study and their isolated places and the time are shown in this map here. So we chose two strands in this study to have PepBio sequencing, that is a third generation sequencing. And why we chose P2B and 29951, it is because, as you can see in this table, for P2B it was isolated in 1948, and it is the earliest isolate in this study. And for 29951, it was isolated in 2017, and it is the latest, the most updated isolated in this study. So that is the reason why we chose the two strands to have PepBio sequencing, and we try to make them have the best genome assembly, especially for chromosome level. So here, as you can see here, for the number of the scaffolds, first, the targeted number of the chromosome of Pyricularia oryzae is 7 chromosomes, and as you can see here, the 7 scaffolds can make the Pyricularia oryzae or P2B, and without any ambiguous nucleotide here. So it is a very perfect genome assembly, and it is a chromosomal genome assembly. For 29951, even though it has 17 scaffolds, however, only 7 scaffolds can account for over 90% of the genomes. So it is also a very good genome assemblies, and both the genome assemblies with the PepBio sequencing, they can belong to the chromosomal level assemblies. And then we try to compare the genome composition with the original reference genome code 1715, as I mentioned before. In the comparison with the reference genome 1715, which is labeled in the color of orange, in the figure A, and is a comparison with P2B. As you can see that there is some translocation between the composition of the genomics in the several chromosomes, and it is actually 4 out of the 7 large chromosomal translocations could be found in P2B with 1715, and for 29951, it is much more syntonic to 1715, so its composition is more similar to 1715. So later, based on the genome assemblies we obtained, here we can conduct the evolutionary analysis in the following approach. So with over 5,000 single copy genes by Rexamo showing that P2B and 2951 are belonging to distinct clades, and that the collected strands were also distributed in a more scattered way instead by geology or time, as you can see and match back to this map. They don't have any relationship or correlationship between the isolated time and the isolated place. And for the right figure here, we try to find the well annotation. The strands have with well annotations released from NCBI genome database, and try to do the comparison. The phylogenetic tree is composed of the strands with what I mentioned, and you can see here P2B and 2951 assembled in this study. They belong to the same clade, and they are also in the clade called Pyricularia oryzae, and also because their hosts are oryzae stava, which is in general is rice. And then we try to conduct the analysis of pangenome. So variation of ortholog groups in the pangenome and the core genome with extrapolated genomes, in which pangenome size of the red line here in this figure in the red line represents an open pangenome because it is a divergent line and instead a converged line. So besides, as the core genomes are the main genome, are the genes that all of the other genes that all of the strands have them, and for the accessory genes are the genes that some of the strands have them but some others don't have the genes. And for the private genes is that only one of the isolates in our study has this kind of genes. So they represent around 60% and 30% and 10% of the targeted Pyricularia oryzae genomes in this study. And so it indicates that the high diversity within this species, and I have to emphasize that within this species, so this is really a high diversity. So in summary, we generated two high-quality genome assemblies to serve as reference genomes of Pyricularia oryzae in addition to the original reference genome released in NCBI database called 1715, and which was firstly released in 2005 and by shotgun sequencing. And so we provide a novel perspective of the genomes, which called P2B and 29551 respectively. So with all the assembled genomes in this study, we also found that the open pangenomes and such open pangenomes suggest that the high genomic diversity within the species. And based on the above suggestions and the reference, we can assume that the fungal pathogen has been undergoing rapid evolution, and based on the results that we have for now, we can then have further research. Especially, we are going to approach the effector gene turnovers, because based on the genomics information that we have for now, we can find the known effectors, and we try to find the gene gain and the gene loss about the known effectors between all of the isolates or called strands in this we collected in this study. So that will be the next step and the next aim for our study here is the effector genes turnover. So here is the topic of my research here, and thank you for your listening.